Okay, so this video is going to focus on specific immunity. So this is going to be that antigen antibody fighting particular pathogens like certain types of bacteria or certain viruses. Okay, not that general response that we talked about in the last video. Okay, so when we're talking about specific immunity, it is also known as our adaptive immunity. So that's just another term for the specific immunity. Again, it is highly specific in its response in that it's only responding to one particular kind of pathogen, okay? But it is able to adapt. It's able to adapt, it's able to change, and it's also able to remember what it has learned, which is key for having a quick response if you're exposed again. So we'll talk about all of this in more detail. So let's talk first about an antigen. Okay, so an antigen is going to be a molecule on the surface of the cell. And hopefully we remember that from blood typing, right? We had type A antigens, type B antigens, and that told us what kind of blood it is. So these are surface markers that are on the cell. And the reason why we have these is they do two things. They both identify the cell as belonging to you, so as self, and they also then identify non-self things. So you have various antigens, and again, the foreign invaders your body will recognize as being non-self, and then your own cells will have antigens that will mark it as self. This is, again, where autoimmune issues come into play, where your body is messing this system up, and things that belong to you, it is actually saying are non-self, and it starts attacking yourself. So let's look at some different kinds of immune cells. So the first one we're going to look at are called antigen-presenting cells. Antigen-presenting cells are also called MHCs. Okay? These are the types of cells that fall under this category are these macrophages, those big giant cells that eat things. So macrophages, dendritic cells, and B cells all can be antigen-presenting cells. And if you look at the image over here on the right, okay, that is kind of walking you through that process. Okay, so this is an APC over here on the right. And again, so this could be a macrophage, it could be a dendritic cell, or it could be an actual B cell. So the first thing that needs to happen is we're going to have phagocytosis of the antigen, of the foreign enemy cell. Right, so that's happening right here, that red rod-shaped cell, that is the antigen that's being consumed. Okay, and once the antigen is taken into the APC, you'll notice it gets broken down into little pieces. That's all of what steps 2 through 4 are, is that the lysosomes within that APC are breaking that antigen down into its little pieces. And once those antigen pieces are broken down, then the APC, the antigen presenting cell, can do just that. It can present the antigen to your other cells. So it was able to realize that this was a foreign invader, and then it basically will chew up the foreign invader and then spit it back out and run around and show that to everybody. Hey, look at this. This is foreign. Hey, this doesn't belong to us. Hey, you need to learn about this so you can start attacking it. Okay, so that's really your main job of your APCs, is they're going to consume those antigens, and then they're going to teach other cells about them. And they're going to run around and they're going to tell the other cells you've got these foreign things going on. Some other type of immune cells are your lymphocytes. And for lymphocytes, we have two basic kinds. We have B cells and T cells. And we'll talk about each one of those individually. Okay? These lymphocytes are screened in the beginning. Okay? They are screened for a self-response. Basically, you're looking to see if they respond to your cells, okay? And if they do, your body will destroy them, so that hopefully trying to avoid autoimmune issues. Your body will trigger that apoptosis response so that it will destroy those lymphocytes. Now, some of them make it through, obviously, but that is one of the systems your body has in place to try to prevent them from attacking your own cells. They have lots of receptors. They're all going to be for the same antigen, though. So that's what it means by being antigen specific. So if I have a B cell, it can only fight or make antibodies for one particular antigen. I need a whole different B cell if I've got another antigen that comes along. So you've got lots and lots and lots of different kinds of lymphocytes so that they can recognize lots and lots of different kinds of antigens. They have the same basic response. The first step that happens is going to be the clonal selection. This is the replication. This is where they make a whole bunch of copies of themselves so that they can do their function. Okay, and then the second step will be that they will activate into either effector cells or memory cells. The effector cells are going to actually carry out the response and fight the antigen, and the memory cells are going to stay behind so that if you have a second exposure, they can help take care of it very quickly. Again, this is the generic response that both B cells and T cells are going to do, and we're going to talk specifics about each one of those kinds in just a moment. On your cells, you're also going to find surface markers called MHC proteins. MHC protein stands for major remember histo is tissue, compatibility proteins. And you have two basic kinds of these. You have class 1, which are on the surface of all cells that have a nucleus. So these would not be on the surface of bacterial cells because bacterial cells do not have a nucleus. So these are going to be but they're going to be on the surface of every single kind of cell that does have a nucleus. These are going to display self-antigens. So these MHC complexes, MHC class 1 in particular, is how you're going to know that this cell belongs to you. You can see a class 1 MHC molecule on this infected cell here, so you know it's one of your own cells, okay, but you can also then determine that it's infected. And you can do that because the class 1 MHC will also display abnormal antigens that are in the cell. So if the cell, for instance, this cell that's infected, you can see it displaying that foreign antigen on that MHC1 complex. Okay? If the cell is becoming cancerous and it's making cancerous proteins, it will also display those. And therefore, your passing T cells, passing B cells, they can see this and maybe attack it. And we'll talk about what this cytotoxic T cell is actually doing here. But this cytotoxic T cell is actually going to kill that infected cell. That infected cell is no good to you. Okay? It doesn't recover from being infected. And so that cytotoxic T cell, one of its jobs is to kill that infected cell so that it cannot continue to reproduce and spread that antigen further. The class 2 MHC proteins, they're going to ingest and display foreign invaders. So these are not doing self. So this cell right here, okay, on the right, this cell in picture B here, it has MHC1s on it. You're just not seeing it in the picture. So does that helper T cell? It has MHC1s on it because all of your cells that have a nucleus are going to have this on there. But the class 2 MHCs, these are going to be on APCs. These are going to be on these cells that ingest or take in these foreign antigens, and then they can present them back out. Okay, so this is an APC. You can see it labeled right there in the top. This APC has taken in this foreign invader. It's broken it down, and it's using its class 2 MHC molecule to help spread the word about this foreign invader. It's showing it off to help activate the other cells. Okay, so we're going to shift and we're going to talk about some specific types of immunity, which are also going to involve these specific types of cells, these B cells and these T cells. So the first one is humoral immunity. Humoral immunity is also called antibody immunity. This is what most people think of when they think of immunity, is that we're making antibodies for things. Okay, so this is very effective for pathogens that don't take over a cell. So this is very effective for pathogens that are found outside of the cell, okay? Because this is going to go around and try to neutralize or destroy those. And so this is not the kind of immunity like we just saw where it destroys cells that are infected. Okay? So basically your first step here is that the B cell is going to encounter the antigen. Remember, a B cell could be an APC. So it could encounter the antigen on its own. It also could encounter it from another APC. But the B cell is going to encounter the antigen. Okay? It's going to bind to its antigen receptors. Remember, they're specific. So this particular antigen binds with this particular B cell. So the B cell encounters the antigen, there's step 1, and then that's going to cause clonal selection to occur. Clonal selection is this replication right here. Okay, so that pathogen binding to it activates the B cell, and that makes it replicate. So we make bunches and bunches and bunches and bunches of B cells. Okay, and so those B cells will replicate, will do the clonal selection, and then ideally that B cell can present to a helper T cell. Okay, so this is that B cell presenting to a helper T cell. And a helper T cell releases substances that are called interleukins. Remember, leuco is white. Okay, so interleukins between white blood cells. So these are basically what the helper T cell does is the helper T cell activates more B cells. It goes and it activates more B cells. And so this will help the B cell fully kick into gear. Okay, and it will help differentiate which ones become plasma cells versus which ones become memory cells. Plasma cells are going to be the actively fighting ones, or the effector cells. So they're going to be the ones that are actively producing antibodies. Okay, that should say effector. That's a big mess. The memory cells are going to be the ones that stick around, and they'll basically remember the infection so that you can fight it again later. And we'll talk a little bit more about those when we talk about primary versus secondary response. But these helper T cells are going to do just that. They're going to activate more B cells. They're going to activate more of them. And they're going to activate some of these cytotoxic T cells by releasing the interleukins. So again, that's the chemical that they release that the helper T cell releases. Right? These are from the helper T cell that they release to help them activate these other cells. Hey, if you guys have Netflix, there is a show on there, and yes, I know it's for kids, okay, and we actually would watch it in class, but we're not in class. Okay, there's a show on there called Storybots, and it's in season 2, and there's an episode about catching a cold, and again, it's in season 2. And this is on Netflix, and the show is called Storybots. And it's a really good show in the sense that it doesn't like dumb down the science for little kids. Okay, and it has some really, really good visuals of like how the helper T cell comes and works, and what the B cell does, and what the cytotoxic T cells. I strongly, strongly, strongly recommend you going and watching this 20-minute episode of Storybots. Again, it's in season 2, How to Catch a Cold. It will really help. Like I said, everyone, whenever we've watched it the past 2 years, everybody says it helps understand it. It really helps them understand it. Okay, so like I said, go watch Storybots. But we're going to keep going through this here. But again, go watch Storybots to help get it solidified. Okay, so let's look at what these antibodies do. Again, these antibodies, they come from the B cells, in particular those active plasma cells. Okay, and so these plasma cells make lots and lots of antibodies, which are also known as immunoglobulins. Okay, these are protein-based, so they're usually a Y-shaped protein. Okay, and they'll have standard parts, and then they'll have parts that are specific to the particular antigen. Remember, these antibodies, they don't, like all of you at some point in time have gotten sick, okay? But you only have antibodies in for whatever you got sick for. That's why you keep getting sick, because you get exposed to a different kind of antigen that your body has to learn about first. Okay, so these antibodies, their job is to neutralize the antigens, and they can do it in a variety of ways. So one of the things is that they can occupy the target binding site. So you see this happening right here. Okay, so they occupy the target binding site, and so now it can't bind to cells. Another way that the antibodies can affect the antigens is through a process called precipitation. So usually the antigens are really, really tiny. You can see precipitation happening in this diagram right here. Okay, usually the antigens are really, really tiny, and so they can actually dissolve in the bloodstream, and so they can move through the bloodstream almost undetected. And so the precipitation, if you remember from like chemistry, right, we bring that solid out of the liquid. So that's what happens, is you've got these antibodies that are making these antigens clump together a little bit, so that way they can't dissolve in the bloodstream. They become too big and they can't dissolve in the bloodstream. You also see that agglutination reaction happening. Okay, that agglutination clumps together those antigens again that are on cells or on viral surfaces. Okay, and it makes them really large. So basically precipitation and agglutination are kind of the same thing. It makes them clump together, but the agglutination is making the antigens that are on a cell, like a bacterial cell, or on a viral surface clumped together, where the precipitation is making the free antigens clump together. But either way, we get this clumping together, and all three of those things, the neutralization where it blocks the different sites, the agglutination, and the precipitation, all are going to help facilitate phagocytosis of these antigens, these macrophages being able to come in and eat them and take care of them. Okay, another thing that the antibodies can do is they can activate that complement system. Remember, that complement system makes those membrane attack complexes that poke the hole in the membrane, and so the cell then will eventually lyse and the cell will die. Okay, antibodies also can stimulate inflammation, right? Bringing more white blood cells to the area, bringing more blood to the area, the swelling, the increase in heat, all things that can impact the antigen's function. So our body has a variety of responses that it can have when exposed to antigens. So the first set would be either it's going to have a primary or a secondary response. And the primary response is going to be that first response, as in the first time it's been exposed to that particular antigen. That response is a little bit slower than a secondary response because that's when it's learning about the antigen and it's learning how to make antibodies for it and how to recognize it and how to attack it. The secondary response is any subsequent exposure that your body has to that antigen. It's already ready to go. So that response is going to be faster and stronger, both faster and stronger. If you look at the graph over here on the right, on the y-axis here is where we have our concentration of antibodies. And so our initial first exposure here, I definitely get an increase in concentration of antibodies. Okay, but it takes a while. My time is down here on my x-axis, especially compared to the second time I get exposed. The second time I get exposed, my concentration of antibodies goes up much higher and in a much shorter span of time. So that's why we don't usually get the exact same disease over and over and over again, right? I build antibodies to one particular antigen, and so if I get exposed to that antigen again, then I can easily fight it off, and I can fight it off quickly. Whereas when I get exposed a first time, it takes me longer. Now, keep in mind, we all get, some of us get multiple colds, every year, that kind of thing. Okay, that means that it's a slightly different virus, so a slightly different antigen every time. And that's why you're still getting sick every time, because you have that primary response every time. Okay? Another type of response that we can have is an active versus a passive type of response. Okay? And so an active response is when we are the ones that are actually making the antibodies. Okay? So we are actively producing the antibodies. Where a passive type of immunity, the antibodies are given to us. Like, for example, a breastfeeding mother, she's giving her antibodies to the infant. Okay? Another type of passive immunity would be you receiving a shot that actually contains antibodies. Not a vaccine where you make antibodies for it, but if you give something that already contains antibodies that were made, that would be passive immunity. And then our last set here is natural versus artificial immunity. And so natural immunity was created by a person. Okay? So whether it was you or another person, but it was created by a person. So for instance, the breastfeeding example we gave for passive, that would be a form of natural passive. The antibodies were made by the mother and then passed on to the child. Where an artificial response here is that the antibodies are synthetic. So for instance, a antivenom. You got bitten by a snake and you need the antivenom. So the antibodies were made synthetically and they were given to you. So that would be artificial passive. Okay, so you can kind of combine these different responses too. So the last thing we're going to look at here is the cellular immunity. Way back in the beginning, we talked about having B cells and T cells, and so the cellular immunity is going to focus on the T cells. So this is what is also called our cell-mediated immunity, meaning that it is controlled and run by the cells. Not the other one was humoral immunity because it's done all in the bloodstream, in the humor. So for our T cells here, we've got basically two types of T cells. We have the helper T cell and we have a cytotoxic T cell. So the helper T cell is going to be activated by those APCs, right? Those antigen presenting cells. So those antigen presenting cells will use their class 2 MHC. Remember that major histocompatibility complex, and the class 2 is the one where they show the foreign antigens. The class 1 is where they show their self antigens. And so a helper T cell, to get activated, needs to do what we call a double handshake. And so by this double handshake, it means that the helper T cell needs to bind the MHC2 complex, so to the foreign antigen, as well as to the CD4 protein. The CD4 protein, it makes sure that the source that it's getting the foreign antigen from is authentic. It kind of does a double check with, like, is this really one of your cells? And is it really presenting something that is foreign? So it binds with both of these instead of just one. So it binds with the CD4 protein and the class 2 MHC. And then that helper T cell is going to go off and it's going to activate the cytotoxic T cell and the B cells. Remember, the B cells are who make the antibodies. So it'll help activate those B cells into the plasma cells that can make the antibodies. And then it will go activate cytotoxic T cells as well. When cytotoxic T cells are activated, then they are actively destroying those antigen displaying cells. So those cells that are infected, the cytotoxic T cells, cytocell toxic, that's poison to the cell. So they're going to cause apoptosis or release perforins, those things that can make those little holes in the cell membrane, which will kill a cell. So they'll either cause apoptosis or release perforins that'll help explode the infected cells. So these are our cytotoxic T cells. Sometimes they're called killer T cells. Both of these cells, both helper T cells and cytotoxic T cells, develop memory cells as well. So remember, with our B cells, we had some that would activate into plasma cells and some that would remain as memory cells. T cells do the same thing. Some of those cytotoxic T cells will become memory cells, so they'll stick around, and some of those helper T cells will also become memory cells. So they'll stick around to help that secondary response be really, really fast. You also have suppressor T cells. Suppressor T cells are going to help kind of mediate and calm down that cytotoxic T cell response. We don't want that cytotoxic T cell response going on for too long or being too strong. We don't want it killing cells that it shouldn't kill. We don't want it killing cells after all the infected ones are gone. We want to kind of keep that reined in and under control. And so the suppressor T cells will help with that. Again, Season 2, the episode on how to catch a cold, Storybots, it has, again, great, like you kind of just see it in action. It gives a little cell that's shaped like a T that runs over and turns on a bunch of other cells. Like, it just really kind of gives you a great visual to help kind of wrap all of this up. Okay, so if you have any questions, we'll be able to answer those in our classes, and I'll be giving you obviously some practice work with this stuff.